So we are today going to discuss about biomolecules, nucleosides and nucleotides, right? So what are nucleosides? These nucleosides are the molecules. They are molecules formed of a nitrogenous base, a nitrogenous base, and a pentose sugar, and a pentose sugar. So nucleosides are the molecules formed of nitrogenous bases and pentose sugars, right? So what are basically nitrogenous bases and pentose sugars? You have studied about pentose sugars. Now I am going to tell you about nitrogenous bases. People are joining. Let me allow them first. Okay. So these nucleosides, along with phosphoric acid, they serve as the building blocks of nucleic acids. So now nucleoside, they are formed of nucleosides, they are formed of nitrogenous base and a pentose sugar. These nitrogenous bases, they are of two types. Nitrogenous base is a nitrogen containing compound. Nitrogenous base is a nitrogen containing compound. There are of two types. We classify them into purines and pyrimidines. We classify them into purines and pyrimidines. These purines, they are dicyclic. They are dicyclic. That means they have got two cyclic rings. They are 9-membered, having nitrogen at position. Having nitrogen at position 1, 3, 7, and 9. Right? So purines and pyrimidines, they are the two types of nitrogenous bases. Purines, they are dicyclic. They are formed of two cyclic groups. They are 9-membered. They have nitrogen at position 1, 3, 7, and 9. Purines include adenine and guanine. Adenine and guanine. They are the only two purines. They are the only two purines. Whereas pyrimidines, they are three in number. One of the pyrimidine is cytosine. Another is uracil. And another is thymine. These pyrimidines, they are also nitrogen containing compounds. They are also nitrogen containing compounds. Pyrimidines are also nitrogen containing compounds, but they are monocyclic. They are monocyclic. That means they have got only one cyclic group. They are coming late, they are joining late to the class. It's disturbing me. So they are monocyclic, they are 6-membered, and having nitrogen only at position 1 and 3. Right? So today we are going to discuss about nucleosides and nucleotides. As I told you, nucleosides, these are molecules or these are compounds formed of a nitrogenous base and a pentose sugar. These people are joining late and disturbing me. So nitrogenous bases, they are of two types, purines and pyrimidines. So nitrogenous bases are nitrogen containing compounds, right? They are of two types, purines and pyrimidines. Purines, they are only two in number, adenine and guanine, they are the purines. While as pyrimidines, they are three in number, cytosine, uracil, and thymine, right? These purines, they are dicyclic. They are formed of two cyclic groups, right? They are 9-membered. There is a purine ring and a imidazole ring. They are 9-membered. They constitute 9 members in the rings, and they have nitrogen at position 1, 3, 7, and 9. While as pyrimidines, they are monocyclic, they are 6 membered, they are having nitrogen at position 1 and 3. So this is the difference between a purine base and a pyrimidine base. Both of them are nitrogen containing compounds, right? Both of them contain nitrogen, but the difference is they are dicyclic, they are monocyclic. They are 9 membered, they are only 6 membered. They have nitrogen at position 1, 3, 7, and 9. They have nitrogen at position 1 and 3, right? Now I will show you it in diagram. By the help of diagram, we will differentiate between a purine base and a pyrimidine base. Is it clear? Is it clear? Now by the help of a diagram, now we will differentiate between purine and pyrimidine base, right? Now I will show you the differences between purines and pyrimidine. So first of all, I am drawing the diagram of adenine. So we will discuss first purines. How many purines are there? Adenine and guanine. They are the only two purines. So first we will draw adenine. Okay. Here is H. This is adenine. You can see this adenine. It is formed of two cyclic groups. It has got two cyclic rings. This ring of adenine, it is known as a pyrimidine ring. This is a pyrimidine ring. And this ring is known as an imidazole ring, right? So you can see it has got nitrogen at position 1, this is position 2, this is position 3, 4, 5, 6, 7, 8, and 9, right? So first of all, it is formed of two cyclic rings. This is one cyclic ring, it is another cyclic ring. So it is a dicyclic compound. It is a dicyclic compound. Is it clear? Then it is formed of 9 members. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So it is 9 members. It is 9 members, right? Who is this for entering and leaving? It is dicyclic compound. It is 9 membered, right? Having nitrogen at position 1. This is position 1. This is position 3. This is position 7. This is position 9, right? So this nitrogenous base, it is adenine, right? And this follows in purines. So as I told you, purines are the nitrogenous bases, which are dicyclic, which are 9 membered, having nitrogen at position 1, 3, 7, and 9, right? Similarly, we have another nitrogenous base, that is guanine. You notice this diagram. You have you drawn this diagram? Diagram banai. So these are two cyclic rings, a pyrimidine ring and an imidazole ring. This ring is known as pyrimidine ring. Pyrimidine ring. This ring is known as imidazole ring. Right? So it has got two cyclic rings. That is why we call it as a dicyclic compound. Right? It is formed of 9 members. It is formed of 9 members. 1, 2, 3, 4, 5, 6, 7, 8, and 9, right? So it is 9 membered. It has nitrogen at position 1, 3, 7, and 9, right? So this is a purine. This is a purine. So first of all, you should know about purine, that they are dicyclic, 9 membered, with nitrogen at position 1, 3, 7, and 9, right? Now, similarly, we will see another purine base, that is guanine. We will have a look at the guanine. So this is adenine. Now we will have a look at the guanine. This is guanine, right? You can see, like adenine, it is formed of two cyclic rings, a pyrimidine ring and a imidazole ring, right? So this is pyrimidine ring, this is imidazole ring. It is again 9 membered, 1, 2, 3, 4, 5, 6, 7, 8, and 9, right? It is again 9 membered. It also has got nitrogen at position 1, position 3, position 7, and position 9, right? So these are both purines. Both of them are purine. This is adenine, this is guanine, right? You can see this adenine at position 6, it has got an amino group, and guanine has got this amino group at position 2. So we also call it this adenine as, we can call it as, they are disturbing me. Okay. So this is adenine, this is guanine. Both of them are dicyclic, both of them are 9 membered, both of them have got nitrogen at position 1, 3, 7, and 9. So these two bases, they are the purine bases. So there are only two purines, adenine and guanine. Now you can see this is adenine, this is guanine, right? And you can differentiate between adenine and guanine. In adenine, the amino group is at carbon 6 position, 6, and this, in this guanine, the amino group is at carbon second position, right? So these are the two purines. Now we will have a look at the pyrimidine, right? So as I told you, pyrimidines, they are monocyclic. Should I rub this? Now we will have a look at the pyrimidine bases. As I told you, pyrimidines, they are monocyclic. They are monocyclic. That means they have got only one cyclic group, right? They are 6 membered. In the ring, there are only 6 members, right? And they have nitrogen only at position 1 and position 3, right? So as I told you, pyrimidines, they are three in number. First, we will have a look at this pyrimidine base. Just a minute. NH3K. It is CH here. Good. It is C double bond O. It is like this. So this is cytosine. So it is having only one cyclic, right? It is having only one cyclic ring. Position 1, 2, 3, 4, 5, and 6, right? So you can see there are only 6 members in the ring, right? So it is only 6 members. There is only one cyclic group. So it is monocyclic. It has nitrogen at position 1 and 3. This is position 1 and this is position 3. So this is cytosine. This is cytosine. Now similarly, we have got another base, that is uracil. This is uracil. This is uracil. 1, 2, 3, 4, 4, 4, 1, 2, 3. Right? This is uracil. Again, having nitrogen at position 1. This is 2, this is 3, this is 4, this is 5, this is 6. Right? So you can see it is again formed of a single ring. This is monocyclic, right? It is 6 membered, having nitrogen at position 1 and 3. Similarly, we have got another nitrogenous base, that is thymine. This is thymine. This is thymine, right? Now, what is the difference between a purine and a pyrimidine base? Can you tell me? What is the difference between a purine and a pyrimidine base? Hello? Can you tell me what is the difference between a purine and a pyrimidine base now? I'm not listening to you. Okay, let me tell it. So purines, they are two in number, adenine and guanine. Both the purines are dicyclic. They are 9 membered and they have nitrogen at position 1, 3, 7, and 9. While as pyrimidines, they are 3 in number, right? Cytosine, uracil, and thymine. They are all pyrimidines. These pyrimidines, they are monocyclic. You can see all of them is formed of a single cyclic group, right? Here like this, right? All of them is formed of a single cyclic group, right? And all of them are 6 membered. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So they are all 6 membered and all of them have nitrogen at position 1 and position 3, right? So this is the difference between purines and pyrimidines. Now there are 2 purines and there are 3 pyrimidines. So all of them are the nitrogenous bases. So first of all, we classified nitrogenous bases into purines and pyrimidines. What are the differences between purines and pyrimidines? I hope you have understood them. Okay? Okay. Shall we move ahead? I'm not listening to you. I don't know what is happening. Okay. I'm not getting a response from you. Now these nitrogenous bases, they combine with pentose sugars. Now when a nitrogenous base combines with a pentose sugar, it forms the nucleoside. It forms the nucleoside, right? So we had a look. We had a look at nitrogenous bases. Now we will see pentose sugars. As you know, pentose sugars, there are 2 types. There are 2 pentose sugars involved. One is known as ribose, another is known as deoxyribose. I hope you know the difference between ribose and deoxyribose. You have studied it. Ribose, it is a pentose sugar, like this. It is a pentose sugar. right? It contains 5 carbons. 1, 2, 3, 4, and 5, right? So it is a pentose sugar. Similarly, we have got deoxyribose. It is also a pentose sugar. Deoxyribose is also a pentose. You can see it here. It is again formed of 5 carbon, carbon first, carbon second, carbon third, carbon 4, and carbon 5. So both of them are pentoses. Now, what is the difference between ribose and deoxyribose? Can you tell me? What is the difference between ribose and deoxyribose? Deoxyribose is deficient. Deoxyribose is deficient in one oxygen atom at carbon second position, right? In ribose at carbon second position, there is H and OH group. In deoxyribose at carbon second position, it is deficient in one oxygen atom. So it is H and H only here. O is missing here, right? So that is why we call it as deoxyribose, because it lacks one oxygen atom at carbon second position, right? So this is ribose, this is deoxyribose. Now any one of them, either ribose or deoxyribose, can combine with a nitrogenous base, right? When a nitrogenous base combines with any one of these pentose sugars, it forms a nucleoside. It forms a nucleoside. Now, what is a nucleoside? So we simply say nucleoside is nitrogenous base plus pentose sugar. Which type of nitrogenous base it may be? It may be a purine or a pyrimidine, right? What type of pentose sugar may be? It may be ribose or deoxyribose. If ribose combines with nitrogenous base, it forms the nucleoside of RNA, right? When ribose combines with nitrogenous base, it forms a nucleoside of RNA, right? When deoxyribose combines with a nitrogenous base, it forms a nucleoside of DNA, right? So we find deoxyribose in DNA and we find ribose in RNA. That is why we name DNA as deoxyribose, because it always has a pentose sugar, deoxyribose. RNA will always have a pentose sugar, ribose in it, right? So nitrogenous base plus pentose sugar, it forms a nucleoside. So this nucleoside can be present in RNA or this nucleoside can be present in DNA, depending upon the sugar involved, right? So that is about the nitrogenous bases and pentose sugars. When they combine, they form nucleosides. Now, how do they combine? How nitrogenous base combines with a pentose sugar to form a nucleoside? So we will see that in another diagram. I will make another diagram and I will show you how a nitrogenous base combines with a pentose sugar to form a nucleoside. Is it clear? Okay. Should I rub this? Should I rub this? Okay. Now let us take a nitrogenous base. Let us take adenine. Let's take adenine. Let's take adenine. Right? This adenine, it can combine with. It can combine with ribose, it can combine with deoxyribose. Let's join it with the ribose sugar. Let us join it with ribose sugar. This is a ribose sugar. Right? This is adenine, the nitrogenous base, right? This is ribose, right? Now they will join by the elimination of a water molecule. A molecule of water will be eliminated. This water molecule it will be lost. When water molecule is eliminated, a bond is formed between this carbon first of the pentose sugar and this nitrogen ninth of the adenine, right? And this bond will be known as what type of bond it will be formed here? Which bond will be formed here between ribose and adenine? I have told you when we were discussing about carbohydrates, amino acids, fats, which bonds are formed. So can you name this bond which will be formed here between the sugar and the nitrogenous base? Oh, come on. Which one bond will be formed between sugar and this nitrogenous base? This bond formed here, it will be known as a glycosidic bond. I have told you what is a glycosidic bond. I have told you that glycosidic bond is a formed between a sugar and any other group, which may or may not be a sugar, right? So this is a sugar. It joins with another group. This group is nitrogen containing group. This is a nitrogenous base. So when a sugar joins with any other group, which may or may not be sugar, a bond is formed. This bond will be known as glycosidic bond. Let us eliminate this water molecule. This water molecule will be eliminated here. It will be taken out, right? And these two compounds, they will join. They will join through this glycosidic bond, right? So this adenine plus ribose, when they join through glycosidic bond, this bond is glycosidic bond. So when they join through glycosidic bond, they form a compound. This compound is known as adenosine. And this adenosine is the nucleoside. As I told you earlier, a nucleoside is formed of a nitrogenous base. Adenine is a nitrogenous base, right? Ribose is a pentose sugar. So when nitrogenous base combines with a pentose sugar, it forms a nucleoside. This nucleoside is known as adenosine. This nucleoside will be known as adenosine. Will it be a nucleoside of RNA or DNA? Would it be a nucleoside of RNA or DNA? Can you tell me? Would it be a nucleoside of RNA or DNA? Adenosine. Is it a nucleoside of RNA or DNA? Come on, tell me. I told you earlier, when ribose joins with a nitrogenous base, ribose is only found in RNA. So this will be a nucleoside of RNA, right? Adenosine is a nucleoside of RNA. If we remove adenine and we will replace it by guanine, which nucleoside will be formed? If we remove adenine and we replace it with guanine, which nucleoside will be formed then when it joins with the ribose? If we place guanine in place of adenine, right? Then another nucleoside will be formed. That will be known as guanosine. That will be known as guanosine. I am not able to hear you. I don't know what is the problem, but I am not feeling good. I should get your response. I want response from you. So if we replace adenine, by guanine, then we will get another nucleoside. That will be known as guanosine, right? Similarly, pyrimidines can also join with these pentoses. If in place of adenine or guanine, a pyrimidine base join, suppose this cytosine joins with ribose sugar. If cytosine joins with the ribose sugar, which type of nucleoside will be formed? Then a nucleoside known as cytidine. Cytidine will be formed, right? Similarly, if uracil joins with pentose sugar. If uracil joins with ribose, then a nucleoside uridine will be formed, right? So these are nucleosides of RNA. These are nucleosides of RNA. So as I told you earlier, in the very beginning, when a nitrogenous base combines with a pentose sugar, it forms a nucleoside, right? So we took a nitrogenous base ribose. We combined it with first adenine. So when ribose and adenine join, they form a nucleoside adenosine. Now if we replace adenine by guanine, right, then we will get a nucleoside known as guanosine. If ribose joins with cytosine, it will form a nucleoside known as cytidine. If this ribose joins with uracil, it will form a nucleoside known as uridine. So these are the nucleosides of RNA. These are the nucleosides of RNA, right? Thymine doesn't join with ribose. Thymine doesn't join with ribose. Thymine is only found in DNA. Similarly, if we replace ribose with deoxyribose, okay, we are replacing ribose with deoxyribose. So this is deoxyribose now. This is deoxyribose, right? When deoxyribose joins with adenine, what will be formed? Which type of nucleoside will be formed? If deoxyribose joins with adenine, it will form a nucleoside, right? That nucleoside will be known as deoxyadenosine. That will be known as deoxyadenosine. If deoxyribose joins with guanine, then it will form deoxyguanosine. Deoxyguanosine. If this deoxyribose joins with cytosine, it will form deoxycytidine. Deoxycytidine. Right? If deoxyribose joins with thymine, it will form deoxythymidine. It will form deoxythymidine. And these will be the nucleosides of DNA. So these are nucleosides of RNA and these are nucleosides of DNA. I hope you are following me, you are understanding me, right? Do you understand me? Hello?
Yes, sir.
Why you are not giving me response? I'm not feeling it good. Okay. So a nucleoside is formed of a nitrogenous base and a pentose sugar. Nitrogenous base plus pentose sugar. And nitrogenous base may be a purine or it can be a pyrimidine. You know which ones are purines and which ones are pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, uracil and thymine, right? So if a nitrogenous base joins with a pentose sugar, there are two types of pentose sugars, deoxyribose and ribose. If we take first ribose and combine with it adenine, it will form adenosine. If ribose joins with guanine, it forms a nucleoside guanosine. If ribose joins with cytosine, it forms a nucleoside cytidine. If ribose joins with uracil, it forms uridine, right? Now, if we replace ribose with this deoxyribose, right? Now, if deoxyribose joins with adenine, it forms deoxyadenosine. If this deoxyribose joins with guanine, it forms deoxyguanosine. If this